Sam, and I'm the Global Product Manager for the RT2 Profiler and PCR Arrays. In the next 3 minutes, I'm going to go through a very quick illustration of how you can build a custom RT2 Profiler PCR Array using tools that we already have available in GeneGlobe. To go through and define the custom RT2 Profiler Builder, there's 2 different links that we can use. The first is to memorize QIAGEN.com/myPCRarray. The other thing you can do is to navigate from QIAGEN.com. Here we're ready to go through and we're ready to start building our custom array. We can go through and we can upload our gene list here. We can go through and click our species, which are human, and then we just quickly hit search, and this will start the process to building our arrays. Clicking on search allows us to go through and GeneGlobe starts searching for our genes of interest. Once our genes are all found, we can go down here and start building our play. And then I'm just going to work my way through the menu here. I'll pick the qPCR instrument that I'm going to use. Today we're using a Bio-Rad TFX96. I'm going to select the plate layout. I have a 34 gene panel plus my control, so it's closest to a 48 gene plate. Down here, I'm going to select the important controls that I want. My GDC, PPC, and RTC control are automatically selected. I can add the housekeeping genes that I want to add now, or I can do it at a later step. I'm going to use ACTP, HPRT1, PTK1, GAPDH, and B2M as my normalization genes. Lastly, down here, I have a choice to select whether or not I'm going to use RT2 qPCR primer assays, which are for your traditional protein coding RNA or your mRNA detection. I can go through and I can load in long non-coding RNA assays, or I can use both. I'm going to start by using just the RT2 qPCR primer assays because I'm interested in looking at the mRNA from these genes. Once I go through and hit select, it's going to go through and it's going to take the genes, identify the qPCR assays, and then upload it into our plate configurator. Now that our gene list has been loaded into the plate configurator, I have some nice instructions here that can tell us a little bit of how we can go through and we can move some of the genes around and how we can search for new genes. And here's my plate already loaded into the plate configurator. So you can see here, I have each of the individual genes that I had requested already loaded in here. The color coding is important as the ones in light blue are mRNA coding genes. The ones in darker blue are the housekeeping genes that I selected. And the ones here in red are important controls that we're going to use during our experiment. If I want to go through and add more controls or housekeeping genes, I can go through here and select those. If I want to change the location of any gene, I can just drag it around on the plate if there's some sort of custom layout that I need during the analysis. In addition, I can go through and I can add additional genes to our plate. So I'm actually going to go through here and I'm going to search for a couple of these different non-coding RNAs. So in the end, I'll be looking at both mRNAs and long non-coding RNAs as well. Once I've gone through and I've loaded up this plate, now I'm ready to go through and I can export the plate as an Excel file. I can save the plate to my plate layout, or I can go through and I can add this to cart and order online. So I'm going to go through and I'm going to order this using my credit card. Click on Add to Cart. Once I click on Add to Cart, up comes another screen that allows me to add additional reagents to my cart. So here I can go through and I can add in my master mix. I can add additional plates, and I can add my first strand kit as well as any of the other things that I may be needing. Here I'm ready to go through, and I can check out online or forward this to my favorite sales rep in order to go ahead and to get my final order in. So there we go, making a custom RT2 Profiler PCR Array in about 3 minutes.
QIAGEN.
Sample to insight.